Hello everyone, today, we are going to discuss all about pharmaceutical suspension. Here is the outline of our topic. 1. Introduction to suspensions. 2. Physical properties of well-formulated suspensions. 3. Pharmaceutical applications of suspensions. 4. Formulation of suspensions. 5. Rheology of suspensions. 6. Viscosity modifiers for suspensions. 7. Preservation of suspensions. 8. Physical stability of suspensions. 9. Manufacture of suspensions. 10. Advantages of suspension dosage form, and 11. Disadvantages of suspension dosage form. Let's begin. A coarse suspension is a dispersion of finely divided, insoluble solid particles, that is, the disperse phase, in a fluid, that is, the dispersion medium, or continuous phase. Most pharmaceutical suspensions consist of an aqueous dispersion medium, although in a few instances, it may be an organic or oily liquid. A disperse phase, with a mean particle diameter of up to 1 micrometer, is usually termed a colloidal dispersion, and includes such examples as aluminium hydroxide and magnesium hydroxide suspensions. A solid and liquid dispersion, in which the particles are above colloidal size, is termed a coarse suspension. What are the physical properties of well-formulated suspension? The product must remain sufficiently homogeneous for at least the period between shaking the container and removing the required amount. The sediment, or creaming produced on storage, if any, must be easily resuspended by moderate agitation of the container. The product may need to be thickened in order to reduce the settling rate of the particles, or the creaming rate of oil globules. Any suspended particles should be small and uniformly sized, in order to give a smooth, elegant product, free from a gritty texture. What are the pharmaceutical applications of suspensions? Let's discuss. Suspension can be used as oral dosage forms, applied topically to the skin, eye, or mucous membrane surfaces, or given parenterally by injection. Many people have difficulty in swallowing solid dosage forms, and therefore require the drug to be dispersed in a liquid. Some materials are required to be present in the gastrointestinal tract in a finely divided form, and their formulation as suspensions will provide the desired high surface area. Solids such as kaolin, magnesium carbonate, and magnesium trisilicate, for example, are used for the adsorption of toxins or to neutralize excess acidity. Suspensions of drugs can also be formulated for topical application. They can be fluid preparations, such as calamine lotion, which are designed to leave a light deposit of the active agent on the skin after quick evaporation of the dispersion medium. Some suspensions, such as pastes, are semi-solid in consistency and contain high concentrations of powders dispersed, usually in a paraffin base. Suspensions can also be formulated for parenteral administration, in order to control the rate of absorption of the drug. By varying the size of the dispersed particles of active agent, the duration of activity can be controlled. The absorption rate of the drug into the bloodstream will then depend simply on its rate of dissolution. Vaccines for the induction of immunity are often formulated as dispersions of killed microorganisms, as in cholera vaccine, the injectable version of which is now being replaced in many countries by orally delivered suspensions. Some X-ray contrast media are also formulated in this way. Barium sulfate, for the examination of the alimentary tract, is available as a suspension for either oral or rectal administration, and propyliodone is dispersed in either water or arachis oil for examination of the bronchial tract. The adsorptive properties of fine powders are also used in the formulation of some inhalations. The volatile components of menthol and eucalyptus oil would be lost from solution very rapidly during use, whereas a more prolonged release is obtained if the two active agents are adsorbed onto light magnesium carbonate prior to the preparation of a suspension. How are suspensions formulated? Let's learn. It is first necessary to ensure that the drug to be suspended is of a fine particle size prior to the formulation. This is to ensure a slow rate of sedimentation of the suspended particles. Large particles, if greater than about 5 micrometers diameter, will also impart a gritty texture to the product and may cause irritation if injected or instilled into the eyes. The ease of administration of a parenteral suspension may depend upon particle size and shape, and it is quite possible to block a hypodermic needle with particles over about 25 micrometers diameter, particularly if they are acicular in shape rather than isodiametric. A particular particle size range may also be chosen in order to control the rate of dissolution of the drug and hence its bioavailability. Even though the particle size of a drug may be small, when the suspension is first manufactured, there is always a degree of crystal growth that occurs on storage, particularly if temperature fluctuations occur. This is because the solubility of the drug may increase as the temperature rises, but on cooling, drug will crystallize out. This is a particular problem with slightly soluble drugs, such as paracetamol. If the drug is polydispersed, then the very small crystals of less than 1 micrometer diameter will exhibit a greater solubility than the larger ones. Over a period of time, the small crystals will become even smaller, whereas the diameter of the larger particles will increase. It is therefore advantageous to use a suspended drug of a narrow size range. The inclusion of surface active agents, or polymeric colloids, which adsorb onto the surface of each particle, may also help to prevent crystal growth. Some insoluble solids may be easily wetted by water and disperse readily throughout the aqueous phase, with only minimal agitation. These are the most widely used wetting agents for pharmaceutical products. Surface active agents, or surfactants, possessing an HLB value between about 7 and 9, are suitable for use as wetting agents. How surface active agents work? The answer is that the hydrocarbon chains would be absorbed by the hydrophobic particle surfaces, whereas the polar groups project into the aqueous medium and become hydrated. Wetting of the solid occurs as a result of a fall in interfacial tension between both the solid and the liquid and, to a lesser extent, between the liquid and air. Most surfactants are used at concentrations of up to about 0.1% as wetting agents, and include, for oral use, the polysorbates or tweens and sorbian esters or SPANs. For external application, sodium lauryl sulfate, or SLS, and sodium dialkylsulfosuccinate can also be used. The choice of surfactant for parenteral administration is obviously more limited, the main ones, use being the polysorbates, some of the poloxamers or polyoxyethylene, polyoxypropylene copolymers, and lecithin. Disadvantages in the use of this type of wetting agent include excessive foaming and the possible formation of a deflocculated system, which may not be required. Hydrophilic colloids include acacia, bentonite, tragacanth, alginates, xanthan gum, and cellulose derivatives. They will behave as protective colloids by coating the solid hydrophobic particles with a multimolecular layer. This will impart a hydrophilic character to the solid and so promote wetting. These materials are also used as suspending agents, and may, like surfactants, produce a deflocculated system, particularly if used at low concentrations. Materials such as alcohol, glycerol and glycols, which are water miscible, will reduce the liquid-air interfacial tension. The solvent will penetrate the loose agglomerates of powder, displacing the air from the pores of the individual particles, so enabling wetting to occur by the dispersion medium. Having incorporated a suitable wetting agent, it is then necessary to determine whether the suspension is flocculated or deflocculated, and to decide which state is preferable. Whether or not a suspension is flocculated or deflocculated depends on the relative magnitudes of the forces of repulsion and attraction between the particles. Deflocculated systems have the advantage of a slow sedimentation rate, thereby enabling a uniform dose to be taken from the container, but when settling does occur, the sediment is compacted and difficult to redisperse. Flocculated systems form loose sediments, which are easily redispersible, but the sedimentation rate is fast, and there is a danger of an inaccurate dose being administered. Also, the product will look inelegant. After the addition of the wetting agent, is to ensure that the product exhibits the correct degree of flocculation. Underflocculation will give those undesirable properties that are associated with deflocculated systems. An overflocculated product will look inelegant and, to minimize settling, the viscosity of the product may have to be so high that any necessary redispersion would be difficult. Controlled flocculation is usually achieved by a combination of particle size control, the use of electrolytes to control zeta potential, and the addition of polymers to enable cross-linking to occur between particles. Some polymers have the advantage of becoming ionized in an aqueous solution, and therefore act both electrostatically and sterically. These materials are also termed as polyelectrolytes. In many cases, after the incorporation of a non-ionic wetting agent, a suspension will be found to be deflocculated, either because of the reduction in solid-liquid interfacial tension, or because of the hydrated hydrophilic layer around each particle, forming a mechanical barrier to aggregation. The use of an ionic surfactant to what the solid could produce, either a flocculated or a deflocculated system, depending on any charge already present on the particles. If particles are of opposite charge to that of the surfactant, then neutralization will occur. If a high charge density is imparted to the suspended particles, then deflocculation will be the result. It is necessary for the suspension to be converted from a deflocculated to a partially flocculated state. This may be achieved by the addition of electrolytes, surfactants or hydrophilic polymers. The addition of an inorganic electrolyte to an aqueous suspension will alter the zeta potential of the dispersed particles, and if this value is lowered sufficiently, flocculation may occur. The most widely used electrolytes include the sodium salts of acetate, phosphates, and citrates. The concentration chosen will be that which produces the desired degree of flocculation. Care must be taken not to add excessive electrolyte, or charge reversal may occur on each particle, so forming, once again, a deflocculated system. Ionic surface active agents may also cause flocculation by neutralizing the charge on each particle, thus resulting in a deflocculated system. Non-ionic surfactants will, of course, have a negligible effect on the charge density of a particle, but may, because of their linear configurations, adsorb onto more than one particle, thereby forming a loose flocculated structure. Starch, alginates, cellulose derivatives, tragacanth, carbomers, and silicates are examples of polymers that can be used to control flocculation. Their linear branch chain molecules form a gel-like network within the system and become adsorbed onto the surfaces of the dispersed particles, thus holding them in a flocculated state. Although some setting can occur, the sedimentation volume is large and usually remains so for a considerable period. Care must be taken to ensure that, during manufacture, blending is not excessive, as this may inhibit the cross-linking between adjacent particles and result in the adsorption of each molecule of polymer onto one particle only. If this should occur, then a deflocculated system may result, because the formation of the hydrophilic barrier around each particle will inhibit aggregation. A high concentration of polymer may have a similar effect if the whole surface of each particle is coated. An ideal pharmaceutical suspension would exhibit a high apparent viscosity at low rates of shear, so that, on storage, the suspended particles would either settle only very slowly or, preferably, remain permanently suspended. At higher rates of shear, such as those caused by the moderate shaking of the product, the apparent viscosity should fall sufficiently for the product to be poured easily from its container. The product, if for external use, will then spread easily, without excessive dragging, but must not be so fluid that it runs off the skin surface. If intended for injection, the product should pass easily through a hypodermic needle, with only moderate pressure applied to the syringe plunger. It would then be important for the initial high apparent viscosity to be reformed, after a short time, to maintain adequate physical stability. A flocculated system partially fulfills these criteria. The product then shows the time-dependent reversibility of this loss of structure, which is termed thixotropy. A deflocculated system, however, would exhibit Newtonian behavior owing to the absence of such structures, and may even, if high concentrations of disperse phase are present, exhibit dilatancy. Although a flocculated system may exhibit some thixotropy and plasticity, unless a high concentration of disperse phase is present, it may not be sufficient to prevent rapid setting, particularly if a surfactant or an electrolyte is present as a flocculating agent. In these cases, suspending agents may be used to increase the apparent viscosity of the system. At low concentrations, many suspending agents can be used to control flocculation, and it must be realized that, if larger quantities are to be used to enhance viscosity, the degree of flocculation may also be altered. These materials are most widely used for the modification of suspension viscosity. Polysaccharides. These include acacia, tragacanth, alginates, starch, and xanthan gum or keltrol. Water-soluble celluloses. These encompasses methylcellulose, having trade names Celicol and Methicel, hydroxyethylene cellulose or Natrosol, carmelose sodium or sodium carboxymethylcellulose, and microcrystalline cellulose or Avasol. Hydrated silicates. These incorporate bentonite, magnesium aluminium silicate or VGAM, and hectorite. Carbomers or carboxypolymethylene. It involves colloidal silicon dioxide or aerosol. It is essential that suitable preservative be included as part of a suspension formulation, particularly if naturally occurring materials are to be used. This is to prevent the growth of microorganisms that may be present in the raw material and/or introduced into the product during use. Some of the natural products, particularly if they are to be applied to broken skin, should be sterilized before use. Bentonite, for example, may contain Clostridium tetani, but can be sterilized by heating the dry powder at 160 degrees Celsius for 1 hour or by autoclaving aqueous dispersions. Because of the complex systems involved and the many factors to be taken into consideration, it is necessary to test the efficiency of a new preservative in the finished product and container by suitable challenge testing procedures. The physical stability of a suspension is normally assessed by the measurement of its rate of sedimentation, the final volume or height of the sediment, and the ease of redispersion of the product. The ease of redispersion of the product can be assessed qualitatively by simply agitating the product in its container. The use of a mechanical shaker will eliminate variations in shaking ability. It is important to ensure that the powder to be suspended is in a suitably fine degree of subdivision, in order to ensure the adequate bioavailability, minimum sedimentation rate, and impalpability. For the extemporaneous preparation of suspensions on a small scale, the powdered drug can be mixed with the suspending agent and some of the vehicle, using a pestle and mortar. It may also be necessary, at this stage, to include a wetting agent to aid dispersion. Other soluble ingredients should then be dissolved in another portion of the vehicle, mixed with the concentrated suspension, and then make up to volume. It is often preferable, particularly on a larger scale, to make a concentrated dispersion of the suspending agent first. This is best accomplished by adding the material slowly to the vehicle while mixing. This stage is important as it is necessary to ensure that agglomerates of the suspending agent are fully broken up. If they are not, then the surface of each agglomerate may gel and cause the powder inside to remain non-wetted. Very intense shearing, however, can destroy the polymeric structure of the suspending agent, and it may be better to use the milder shearing and then allow the dispersion to stand until full hydration has been achieved. This may be instantaneous or may, as with tragacanth, take several hours. If the suspending agent is blended with one of the water-soluble ingredients, such as sucrose, this will also aid dispersion. The drug to be suspended is added in the same way, along with the wetting agent. For every hydrophobic drugs, wetting may be facilitated by mixing under reduced pressure. This has the additional advantage of aerating the product, and thus improving its appearance. Other ingredients should now be added, preferably dissolved in a portion of the vehicle, and the whole made up to volume, if necessary. Finally, homogenization is performed to ensure complete dispersion of the drug and the production of a smooth and elegant preparation. It is also possible, though much less widely used, to suspend an insoluble drug by precipitating it from a solution. This can be accomplished either by double decomposition or, if it is a weak acid or a weak base, by altering the pH of its solution, or by precipitating the drug from a water-miscible solvent on the addition of water. This method may be of use if the drug is required to be sterile, but is degraded by heat or irradiation. A soluble form of the drug is dissolved in a suitable vehicle, sterilized by filtration, and then precipitated to form a suspension. In normal circumstances, aqueous suspensions can be autoclaved, as long as the process does not adversely affect either physical or chemical stability. These advantages can be obtained by formulating API as a suspension dosage form. (1) Suspensions are a useful drug delivery system for therapeutic agents that have a low solubility. Although low-solubility therapeutic agents may be solubilized and therefore administered as a solution, the volume of the solvent required to perform this may be large. In addition, formulations in which the drug has been solubilized using a co-solvent may exhibit precipitation issues upon storage. (2) Pharmaceutical suspensions may be formulated to mask the unpleasant or bitter taste of drug. For example, chloramphenicol. 3. Pharmaceutical suspensions may serve as an alternative means used to administer drugs to children, including pediatric and geriatric patients, and older patients who have difficulty swallowing solid dosage forms. 4. Drug in suspension exhibits higher rate of bioavailability than other dosage forms. Bioavailability is in this order. Suspensions are expected to demonstrate improved bioavailability compared to the same drug formulated as a tablet or capsule. 5. Pharmaceutical suspensions may be formulated to provide controlled drug delivery. For example, as intramuscular injections. 6. Chemical stability of certain drugs, for example, procaine penicillin G, can be improved when formulated as suspensions. 7. Pharmaceutical suspension offers resistance to degradation of drugs due to hydrolysis, oxidation, or microbial activity. 8. Duration and onset of action can be controlled. For example, protamine zinc insulin suspension. 9. When compared to solution dosage forms, relatively higher concentration of drugs can be incorporated into suspension products. The disadvantages of suspensions as pharmaceutical products include: 1. Pharmaceutical suspensions are fundamentally unstable, and for this reason, it requires formulation skill to ensure that the physical stability of the formulation is retained over the period of the shelf life. 2. Aesthetic pharmaceutical suspension is difficult to formulate, that is, they lack elegance. 3. Accuracy of dosage is less reliable than with solution, unless suspension is packed in unit dosage form. 4. Suspension formulations may be bulky, and therefore difficult for a patient to carry. 5. Preparation must be shaken prior to measuring a dose. 6. They have pourability and syringeability issues. For example, injectable suspensions. 7. Crystal formation or Oswald ripening and breaking of suspension may occur. By the end of this talk, we have learned the pharmaceutical suspension in detail. Let us know if you want help regarding any topic. We welcome your feedback and suggestions. See you later.